So now I know all of you have a lot of questions, but we'll have a small panel discussions with the questions that we thought will help you, and then we'll take the questions from the audience. And for that, I would like to invite Dr. Pallavi, Dr. Abhishek, and along with that, our another panelist is Dr. Diane McCarthy. Dr. Diane is the Senior Director of Science and Standards in USP's Global Biologics Department, and she leads the development and maintenance of physical and documentary standards to support the quality of medicines, and she oversees the USP Biologics Laboratory both in the US and India. Her team supports standards and tools across diverse therapies, including peptides, vaccines, cell and gene therapy, monoclonal antibodies, and other protein biotherapeutics. Before joining USP, she worked for several small CROs that focused on characterizing biologics, host cell proteins, and biomarkers. She has earned her PhD in biochemistry from the University of Texas at Austin. Dr. Diane, please welcome on the stage. And our last panelist, Dr. Shashwati Basak. She is Vice President and Head of Cell and Gene Therapy Unit at Intas. And she is leading the development of several programs in both cell and gene therapy, spanning from early stage research to clinical stage. Prior to this, she served as the head of quality and regulatory operations at Immunil Therapeutics, focused on developing CAR-T therapies. Dr. Basak has a PhD in molecular and cell biology from the Indian Institute of Sciences, Bangalore, and she did her 2 postdoctoral fellowship from the Salk Institute of Biological Sciences and Stanford University. She has published her research work in numerous top peer-reviewed international journals and has also served as a reviewer for many American Association of Cancer Research journals. She has over 20 years of scientific leadership and experience and worked in several biotech pharma companies, including Biocon, BMS, R&D Center, Origin, and Immunil. So I invite all the panelists for the panel discussion, please. So I would start with Abhishek and Pallavi. Can you, any one of you can start, can you share what are the main challenges that you face, and what do you think needs improvement in the thought process when people are developing bioassays?
So, hello. Yeah. So the main challenge which I have seen over years of experience is that sometimes assays are initiated without really understanding the nitty-gritties of cell culture. So that is the most important thing. One requirement that every person needs to understand and implement is through sheer observation. Small things like which reagents impact the cell growth, cell density, the cell behavior. These are really small but very important things. For example, when one changes a lot of a particular reagent or even a brand of a particular reagent, although it may claim that it is equivalent, sometimes there are impacts on the cell growth, cell behavior, which are going to impact the outcome of the assay. So these are very important things, and one has to be very observant and critical about these things. The other thing that I can quickly think of is how you devise and develop the method which ultimately leads to what sort of acceptance criteria you propose for a method. If you start off with something that is too stringent, you're bound to look at a lot of method failures. Possibly this might crop up post-validation and lead to a lot of OSs. So it's very important to be very critical about what sort of acceptance criteria you actually develop and propose, because that is actually going to impact the product quality and the acceptance or rejection of your product. So this is my 2 bits on this.
Thank you. Abhishek?
So, Pallavi has covered the developmental aspects, but one of the key things is you need to understand the system. Mechanism of action is one thing, but you need to understand what are the capabilities of your assay by itself. So one is acceptance criteria, but is it detecting the right levels of sensitivity? Is it detecting the right levels of degradation? So you need to sort of qualify or characterize your method from that perspective to understand. And as you gain more and more knowledge through a variety of your product variants or these different sample types, I think that knowledge should get into the final format of your assay along with the acceptance criteria. So that's one thing. Second thing is, a lot of these assays, mostly potency assays, are primarily through engineered cell lines. So how they drift over time is an important exercise, how you need to trend not just your potency values over time, but also potency or activity values over time, but also EC50, even your upper asymptote and lower asymptote. So these are important because that will tell you that your assay system itself is stable and continues to have the same level of sensitivity from the time that you generated this initial assay cell line till the point that it gets commercial and much beyond. So it's a life cycle management of assay. You need to have a ground level understanding. What is happening? What's going on with your system? Is it stable, or when do you not want to use your system beyond a certain cell passage number?
Thank you very much, Abhishek and Pallavi. That was really helpful information that you shared. And we have been talking about mostly monoclonal antibodies, T-cell therapies for vaccines, like how to develop. But I want to also, because Shashwati is here, I also want to talk a little bit about cell and gene therapy. And the developing bioassays for cell and gene therapy is, because it's an emerging field, especially in India, it's much more complex than MABs. MABs we have kind of gotten used to and we are just ironing the wrinkles. But share your thoughts on cell and gene therapy and what challenges you face.
Hi, good morning, everyone.
No.
Okay. So I think since morning we heard 4 talks. Just combine the challenges of all 4 in short. So I'll take the example of CAR-T cells. So what is a CAR-T cell? Let's understand. So I think Dr. Nimesh Gupta very nicely presented on the T-cell assay. In CAR-T cell is the product. It is a genetically modified T-cell. To do the genetic modification, first we need a plasmid which will have the genetic element. With that we make the lentiviral vector. So we have a virus. Using the virus, we transduce the T-cells to have the genetic modification into the T-cell to express the scFv region of a monoclonal antibody. And then we try to put it back into the patient, which is patient specific, meaning for every patient there is a product. So it is not allogenic, it is called autologous in this case. So I think you can now imagine the complexity involved. We are not talking about a small molecule, we are not talking about a large molecule, we are talking about a cell which has to be characterized at so many different levels in order to understand what the functionality is, right? Coming to few of the challenges, I think all the challenges that, as I mentioned, like you expressed in terms of developing or qualifying a potency assay, but I will focus again on the CAR-T cells as an example. So limited material availability. We're talking about autologous therapy. Now, because each patient has to first donate those T cells and those T cells are modified and are put back into the patient, we have very little material to work with, even to develop the assays. So we need surrogate cell lines. So we may have a B cell line, which will be used as the assay template on which maybe healthy donor T cells are modified using the LV that I talked about to introduce the genetic modification to express that particular scFv region, which is specific to the antigen of the B cell, which we are trying to target and kill inside the patient, right? So, so material availability is one. Lot-to-lot variability. While we talk about biosimilars, you have a reference material, right, to which you're trying to show the similarity. Each patient has different subsets of T cells. Like anybody who has worked in immune system would understand that how varied it can be. And there are experts. Dr. Manjula Das is right here. She's an expert immunologist, so she can also vouch for it, like how variable the samples would be, even from healthy donors. So to attain that kind of consistency is also a challenge. What else can I think of? Because it is a cell line based, product is a cell, potency assay is also based on a cell line. So it's like an art more than, we don't have a machine to do anything, right? So the person has to be very well trained to take care of these kind of assays. So I'll stop there and let's hear from others.
So Shashwati, since you covered CAR-T and lentivirus, maybe I'll talk a little bit about gene therapy and focus maybe on AAV. So I will qualify this to say that I'm not personally doing bioassays for AAV, but USP has brought together a number of experts in the field through some roundtables and some stakeholder forums, and we have a chapter currently in development on AAV that will cover partially potency. So I'm kind of bringing those lessons that we've heard from those experts in my comments today. I think, you know, some of the challenges, I mean, potency is one of the hot topics every time we bring people together for AAV because it is so complex. I think there's a couple things that make it unique relative to maybe MABs where it's a little bit more established. One is that I think it's clear right now that you need to have a set of different tests. You know, you're looking at different aspects. You're looking at the infectivity, you're looking at the gene expression, and then you want to see that that gene expression is actually leading to the protein, you know, that you're intending to make in the patient. So, you know, setting up that battery of assays is really important. And I think also, you know, I really like Pallavi's comments for MABs of starting very early in terms of developing those assays. I think what we see more in the gene therapy space right now is that those assays evolve a lot from early phase to late phase. And so, you know, one of the things that you really need to plan ahead for is how are you going to bridge across those assays? Because typically what you're going to use at a clinical stage is not probably what you're using in the early stage. And so I can't emphasize enough that, you know, you need to make sure you have retains so that as you develop those more sophisticated assays, ones that can be used perhaps for release, whereas many of your earlier ones probably aren't appropriate for release, that you have the retains that you need from those early batches and those early assays so that you can bridge across, you know, to your clinical program.
Thank you, Dr. Shashwati and Diane. Dr. Diane, it's good to know that USP is working on a chapter on this, and we already have chapters on bioassay design, development, and validation. We'll be happy to talk about it later on. Now I'll move on to the next section, wherein I wanted to ask our panelists about the statistical part of bioassay, especially Dr. Abhishek and Pallavi. So at what point do you think that we should involve a statistician? Or how heavy statistics is there during your data analysis? When do you start thinking about it, that, oh my God, bioassay, now I have the data, I will analyze it, or now I'm designing an assay and let me discuss with a statistician. So your comments on that.
So I think a bioassay is not a statistics problem. It's not even a scientific problem. It's an engineering problem, right? So once you design an assay cell line, and that's most of the time it gets developed in a way early in the program, then there are two aspects to it. One is what is the assay design. That's very important because that a beautiful looking cell line can give you very variable assays. And in my experience, assay design has played a huge role in calling an assay variable or not. You know, are you placing those replicates right? Are you running a 3-plate assay? You know, are you minimizing the variability causing parameters? So that is one important thing. And yes, statisticians can help in designing it, and I've seen a variety of statisticians when I was working with USP on these 3 chapters, and Shubhrata, we have some private conversations around that. But one of the key things is you need to keep it simple. It cannot be a complex assay design. That's one important part. So how to make it simple? It's more, I would rather have my team, the bioassay folks, talk to application design people, engineering mindset, right? How to make it work. That's the bottom line. You don't have to be purely randomizing it and using this automation to make it work.
Yes.
It can be very simple, alternating, we can talk more about that. Second aspect is the actual data, how to analyze it. So there are 3 parts to it. One is your assay acceptance criteria, what it should be, what should be the parameters you need to put in there. Second is your assay trending, we just talked about that. And third part is your validation. So trending and validation or qualification, those things are established. But in the assay acceptance criteria is where statisticians are heavily involved. Question is, do they need to? Maybe or maybe not. USP has fantastic chapters now which guide them how to go about that. But importantly, I feel it's very intuitive. You need to look at your assay curve, which is a sigmoid curve. When you call it parallel, visually, when it is actually parallel to it. There are 3 parameters, top end, bottom end, and that the middle part. 95% bioscience people think the P parameter is slow, it's not. So you need to just think, visually, if I'm calling this as a parallel, or these 2 curves parallel, the top end should be close to each other, the bottom end should be close to each other, and the center, which is not P parameter, it's a mixture of B, A, and D. Amgen called it tangent, we call it tangent there. So that need to be matching, and once it's matching, your parallelism criteria is set. You can run 20 assays across a period of 3 months and establish what my range I'm observing. So yes, it's important to include statisticians just to validate some of these things, but I'm always of the unconventional opinion that statisticians should not be driving bioassay data. It has to be scientists, and scientists with basic understanding of what I'm looking for, what I should be calling parallelism. That should really make the cut. But yes, I'm not dismissing this fantastic statistician, but I still believe that they have a lot more important problems to solve with bigger data sets that are available elsewhere.
Pallavi.
I completely second what Abhishek has mentioned, and regulators are increasingly emphasizing on presenting data in a very simple way without having too many confounding and complicated statistics involved in it. As far as the question on when the statistics starts, normally we do it in-house, and it would start with something as basic as defining the assay acceptance criteria. But that actually points out to the robustness of how you go about with your method development. Because if you have too many variables in that, obviously your acceptance criteria are going to be all over the place. And most important is to start trending the data right from the first experiment that you execute for method development. And once you have a fair control and understanding over your method, that's when you actually start looking at the data holistically. That is an ongoing process that will be starting from your development till the point your method gets not only validated, but you also have data from manufacturing batches coming, and that's how you're going to look at potential drifts in the assay parameters. It's very important to have appropriate QCs, and regulatory guidelines also now emphasize on the role of having standalone QCs. So if you go through the recently approved products, they would look at all of these parameters in toto. So in addition to what Abhishek mentioned, yes, your 4PL is extremely important, but you don't need to overcomplicate it. Look at simplistic ways of looking at the same data, analyzing it in a way that essentially proves the capability of your assay and also does not raise any questions about your product performance. Like a case in point, if you have too many complicated acceptance criteria, there is a chance that your method will have high rates of failures. So once you develop a method, think about projecting your acceptance criteria, keeping the product quality in mind. While it looks very fancy to have lots of statistics applied to a method, you need to understand how that actually correlates with what is the expectation from your method. That's where the concept of analytical target profile comes, right? How is it linked to what critical quality attribute you're looking at? And what are you trying to establish by using that method? So if you have these points in mind, then you would very well understand how much of statistics and where you need to apply, right? Wherein you can possibly, if you look at the recently approved analytical similarity packages, agency is not in favor of tiering the critical quality attributes. They are okay with mean plus minus SD approach. So we need to understand that this is something that's a top-down thing, right? It cannot be that I accept analytical similarity for a method wherein my acceptance criteria are very complex. So we need to look at the entire thing very holistically. So that's what I would put.
Can I add a comment, Shubhrata?
Quickly.
Just to validate it, I'm not a great cell biologist and all biologist. So the whole career that I made in bioassay world is primarily working on the design aspect, which is not complicated. I can keep it simple and ensuring that the statistics that we are applying continues to be minimal, because the real power is your system, the assay you are running. It's not in the stats. Stats is just final touch to make sure your assay goes similar.
Yeah, very well said, Pallavi and Abhishek. So we have to understand what we are doing and keep it as simple as possible, not include too many criterias, because end of the day when you are doing that Excel sheet, everything fails and you are really upset and you cannot do another assay. So, yeah.
So, before you
Yeah.
close, I know I've been exceeding the time consistently, but
ahead,
just
go
what Abhishek alluded to some time back, that is where the design of your method and the design of your qualification and
Yes.
validation becomes very critical.
yeah.
What sort of samples you use to show the capability of your method, that is directly going to correlate with your method performance. That's going to help you to trend and understand the stability data. You're going to understand the product degradation behavior. That's going to have impact on assigning shelf life and other attributes. So it's not, you don't need to look at the method per se. You have to look at the entire program, the development program, and the way the product will move into approval. That's how it's supposed to be done.
It's very difficult to ask bioassay scientists to stop. So I will move on to my next section, which is we'll talk a little bit about reference standards. But first we'll start with the perspective of cell and gene therapy, because as you shared, that it's very complex, very personalized. Now, where do reference standard even fit in? Will they fit in? And can you share like what kind of standards will help the industry? So from your experience, let us all get benefited.
Right. So I think I will talk about two different aspects here, two different products here. One, the cell-based but genetically modified therapies, and the gene therapies. So in the autologous cell-based therapies, it's very difficult to get a reference standard. What is the reference? The patient is the reference, right?
Right.
So I think in the absence of any kind of reference standard, the best one could have is internal reference material. So which could be a clinical indication specific cell line, which we are trying to cure in the patient or at least treat in the patient. So having that cell line, having control over the variability of that cell line over number of passages that one can use. So if we can control that kind of variability, like to what extent we should grow them, what is the cell number at which we should do a certain assay and all that stuff. So that's probably the best that we can get to it. Apart from that, maybe the quantification of cell numbers. So if there are beats for flow cytometry based assays, for example, so that would probably help in quantification of the cells. There are many antibodies. So flow cytometric methods, I think there are many more which can be controlled. So if each of those assay input materials and the reagents are controlled, I think that would be very good. And if we have reference material for that or reference standards for that. Switching over to the gene therapy products. In case of gene therapy products, if we can have reference materials, say for LV, to determine the copy number. So if there is a cell line which is transduced with the LV, and the LV has integrated into the genome, and we know the copy number, a predetermined copy number, which we can utilize every single time for our assay, for our product. We know at least that our assay is correct because the reference material is giving me the exact same copy number that I expect. So my assay is good to go even for my test material or the product. That is one example. The second one I can give probably for the AAV based gene therapy products, wherein empty versus full. To make it a little bit more complex, I am expecting from Diane and USP partial and overfill too. So I think that would be a good challenge for all the people who are involved in getting these reference materials for different serotypes of AAV, because we have multiple serotypes. So if we can have reference materials, empty capsids, partially filled capsids, full capsids, and overfilled capsids, fully characterized and made available to us, that would be wonderful.
Excellent. Thank you so much, Shashwati. Dan, you want to say something?
Sure, I'll comment just a little bit. So maybe I'll start with MABs, and then I'll come to cell and gene therapy. But I think we are looking at really expanding our portfolio of MAB-based standards, and I think we've heard a lot of interest in getting to some of the functional aspects. So looking at some of the MABs that are commonly developed for potency assays, as well as perhaps for some of the binding assays. So those are in, I would say, fairly early development on our end. Shubhrata and her team here in Hyderabad will be instrumental in developing those, but I'm certainly happy to talk to people offline if you have specific asks or specific areas of interest. On the cell and gene therapy side, quite interesting. So vector copy numbers, I'm glad you mentioned that for lentivirus, and we will have a standard coming for that'll help with that, the consistency of that measurement, hopefully within this year, I would hope. I think we're still finalizing some of the paperwork on that one. And then the empty full question is a great one. I mean, I think it comes up in every single discussion around AEV. I think it's a challenging question because there's a number of different orthogonal methods that can be used. And so we actually embarked a little over a year ago on a collaborative project with NIST in the US, the National Institute of Standards and Technology, as well as another organization you may not be as familiar with, but it's called NIMBLE. It stands for National Institute of Innovation in Manufacturing for Biologics. And so NIST and USP are working under that NIMBLE umbrella to do a multi-lab study of multiple methods for empty full. That actually, the pilot study is now complete. I think we had 6 or 7 labs that were involved in that, and so we're currently crunching data, and we'll be, feeding that back out to the field. But I think it's a challenging question, and I think this study was always intended to support development of reference materials, but really to understand first, like, how do we measure it, and then that'll kind of help us determine what's the most relevant, reference material there.
Thank you very much, Diane. So now I'll ask Pallavi. Pallavi, you do a lot of binding studies, and some of these have no functional assay standards available for. There are few international standards available, but as far as I know, commercial, no pharmacopoeia has. So what is your take on this? What kind of standards will help your assays?
So classically, how we begin with method development would be to use RMP. Then we would move on to have our internally developed and qualified reference standard. It's a preempt that has to be as thoroughly characterized as possible. By then, we would have also established the QTTP using some of the RMP batches that would be available with the organization. So typically, how it starts is with RMP procurement. Then we would do some QTTP analysis. Then we would go on to develop and characterize our internal reference standard, and that would be used subsequently for all our method development experiments. So we already have a reference point. We have our own in-house material which shows the similarity with the RMP, and that is typically how it is as per the guideline expectations as well.
Okay, thank you. So Abhishek, I have one last question for you. Is that at what stage should people incorporate a reference standard, a potency assay standard? Suppose I'm developing a potency assay, and I will have to compare with something. So either it's RMP, but if a RS is available, at least I advocate including an RS, but what is your view on that?
So I think for biosimilars, it's easy. You have a reference product available. So it has to be from the very beginning just to make sure the product you are making matches. For novels, in the discovery stage, when these candidates are being identified, that's the right time to understand what the EC50 of your the product that you're making, the final lead candidate, 1 liter or 5 liter scale product, how it is giving. And then initially that's called a primitive or preliminary reference standard that can be used to take it beyond IND and make the product consistent as compared to that primitive reference standard. So it has to be way early in the program so that you ensure that those PQAs, product quality attributes, don't change over time. And it is not just applied to potency assays, but also to other binding or physicochemical attributes. So way early in the program, it's not just advisable, it's highly recommended you do that.
Thank you. So what we learned is using a qualified material way early in the program, keep it simple, do as many orthogonal assays as possible, and statistics is important, but again, learn the process and consult when required. And Shashwati gave us a lot of insights on cell and gene therapy. Diane told us about reference standard. So I will open the floor for question answers. You can ask all the speakers. Diane, would you want? You can stay if people have questions for you. I mean, it's up to you. Okay. Yeah. So if anybody has questions, please.
So it is really nice panel discussion, and I got a lot of updates on the current trends on the cell-based assays. So mainly, I just wanted to understand the from the Abhishek or maybe from Pallavi. What is your thought process on assay acceptance criteria and parallelism parameter at what stage of the assay? Because we have in a multiple stages. What I understood is that a development stage, then you will do the optimization, then you will do the qualification of the method, and then finally you will do the validation when you transfer the method from R&D to QC. So these are the multiple stages of the cell-based assays. So parallelism is one of the very key system suitability parameter, or would be the acceptance criteria. So parallelism could be included at what time actually? During the development, during the optimization, and like that.
So it captures everything. I was just telling Pallavi. So there are two levels, as I always see it. One is the basic assurance that your assay is capable, like you're keeping it very simple. So your acceptance criteria at that level needs to be very absolute. That, initially my first assay that I got from R&D, it has this level of system suitability in there. That is comparing to the reference standard. Your sample needs to be, let's say, within plus minus 30%. So that's the basic first level of it. As you gain more data, then you understand your assay capability, and that gives you the final second level of assay acceptance. So that's a statistical way of tightening your criteria based on your actual available data. So two levels. And it should be from the very beginning. Level one should be from the very beginning. To your first question, that system suitability versus sample acceptance. So assay suitability is key, because you need to know if your assay is, or your system is indeed suitable to measure what you are intending to do. So that needs to be very self-contained in terms of what parameter you'll be tracking to ensure that the assay is there. So assay control is usually one of the extra lots that you generate. Not early in the program when you have just one particular lot. However, as you generate another lot, just to show consistency, you can apply that as assay control from the very beginning, because that will give you a good understanding of my assay control implementation. In my experience, not many companies focus on assay control per se, and because of that, they have to fail the assay. They result in out of specs later in the game. So putting an assay control and its parallelism criteria or its potency itself in your system suitability, that will prevent a lot of heartburn. You don't have to defend out of specs, and it will be a great assay to begin with. So early in the program, but just be phase appropriate to make sure that it's not causing unnecessary burden as you generate and gather more and more data around your assay. So phase one might be the right time.
I would also want to add one thing. Final acceptance criteria for any method are expected to be derived only after your method is completely qualified. So till that time, it is open to develop, redevelop, revisit your method and the acceptance criteria as many times as you want to. But the underlying philosophy is that the first time you do it, you would have a fair understanding about the method itself. So you don't need to, basically reinvent the wheel time and again.
So qualification is an end of the
Qualification would be the end of the activities as far as R&D is concerned. But you can still have challenges coming from transfer as well as validation, and also from the manufacturing data.
So till that time you can keep as in monitoring of these all.
So certain parameters have to be monitoring parameters, and certain parameters have to be stringent assay acceptance criteria.
So Pallavi, I have to stop you. People are waiting for the next session, so we'll give opportunity to others if anybody else has questions. Yeah, please go ahead.
My name is Sheetal Raut. I am from 𝐂𝐞𝐥𝐥 Laboratory. In continuation with that, I just would like to understand that we develop an assay in R&D and it is well qualified with whatever parameters. But we do see a lot of variation when we transfer the assay to QC. A lot of variation in DRC and a lot of variation with system suit failure. So what do you think, Dr. Pallavi and Dr. Mathur, what do you feel, what are the critical parameters which we should be emphasizing while doing method transfer to QC?
The most critical parameter is communication. Right? That's my experience time and again. The understanding has to be same on both sides. And the way we draft documents is equally important. So it cannot be left to the assumption and the understanding of the language. Rather, it has to be drafted in a very clear, very affirmative language such that every bit of detail which is going to impact your method needs to be captured appropriately. That is why in my slide deck also there was a mention about having appropriate documentation, right? Several examples in my experience wherein it's only the matter of difference in understanding that led to assay failures. Okay, QC and R&D operate on very different modes. So we need to factor that point right in the beginning, need to have certain prerequisites to be ticked off a checklist so that there is certain degree of harmony in the understanding to begin with. And if you look at the regulatory guidelines, they also expect that there should be some sort of pre-discussion or there should be some sort of checklist, that both parties need to address and come to understanding. That's one thing. As I mentioned in the very first question of the panel discussion, challenges in bioassay, very typical because it starts out with a lack of understanding, or I would say a different understanding, to put it in a different way. People who have probably handled cells in and out during the phase of method development have many soft learnings which don't get translated when they transfer the method. And that's where a lot of challenges can actually be abrogated. So there is no one solution for this, but the only thing is technical learnings need to be documented and presented appropriately so that it's a win-win situation. Because more often than not, transfers will proceed smoothly if this is taken care of.
Discuss offline. So one of the key difference between R&D and QC is R&D is testing maybe 2 or 3 or 4 samples at a time. QC maybe 20, 25 samples a day. So that needs to be incorporated and validated in R&D environment. If you run 20 assays, what things change? You have 10 plates lied down in your incubator or in your hood for 15-20 minutes, cells in media, doesn't impact your cells or not. So that is scale which rarely gets tested in R&D environment. I think that needs to be replicated, confirmed, and then that information should go to QC along with other nitty-gritties in there. Again, we can talk more because there's a lot of heartburns, I think, all around.
Yes, sir. Yes, sir.
R&D. I've worked on both sides. I'm sure Pallavi also worked on both sides, so we have